Hello and good afternoon friends, welcome once again to the CEC-UGC live lecture. Dear friends, as you know that we are celebrating Digital India Week. This week has been started from 1st July and would be continuing till 7th July. So all through this week we are just saying that CEC is doing commendable job in imparting e-education which is the proposed plan of Digital India Week. We hope to impart e-education throughout. So dear friends, taking our lecture forward, I would like to tell you all that today also we are going to talk on nitrogen assimilation. This is the third lecture in this particular series, I would say, and today we are going to discuss on regulation of nitrate reductase, and for this we have again with us in our studios Dr. Ashish Kumar Nandi. Dr. Nandi is a professor in School of Life Sciences, Jawaharlal Nehru University, New Delhi. So dear friends, let's take advantages from his experiences and try to understand more about nitrogen assimilation. So first of all, I would like to welcome our guest, Dr. Ashish Kumar Nandi. Dr. Nandi, welcome once again. Though we have discussed a lot about nitrogen assimilation, and today also we are going to discuss about the regulation of nitrate reductase. So over to you, sir.
Yeah, thank you very much. Indeed, this is a continuation of my last two lectures, in which I have been talking about how plants acquire nitrogen and how it converts into organic molecule. So, in fact, as such in the mineral nutrition, this will be the fifth lecture. In the first two lectures, which is still available through the YouTube, I have discussed what is mineralization and what is assimilation. Again, actually to tell you that assimilation is the process by which an inorganic molecule is converted into organic molecule, and that is the capability which is mostly present in some microbes, and largely it is done by plants only, and for that reason the entire living kingdom depends on the plants. So, again, among the elements, like plants need around 19 to 20 different elements, and not only plant, all the living kingdom depends on about 19-20 elements, and nitrogen is one of the most important element among them. And last two lectures we have discussed how plants use elemental nitrogen for their growth. Nitrogen is mostly present in gaseous form in air, which is non-usable form. So, this nitrogen must be converted into usable form, and plants can acquire in two different forms. One is ammonia and other is nitrate. So, before the elemental nitrogen can be used, there must be mechanism by which this nitrogen converts into nitrate or ammonia. So, in the first class I have spoken how the symbiosis between plant and bacteria helps converting elemental nitrogen into ammonia. And the second class I have discussed the regulation of this ammonia production, as well I have also introduced how the plants use nitrate to make ammonia. So, this nitrate cannot be used until again it converts into ammonia. So, plants absorb this nitrate, converts into ammonia, and I will continue discussing on that the key issues like how this process is regulated and why it should be regulated, and its other details machineries, how this process is carried on. So, if I again give an overview what I told you earlier, that can I have a mouse please? Say this is how the ammonia is used, and this the cartoon in the right shows how nitrate is used. And now coming to the nitrate 2 ammonia. This is a 2-step reactions. The nitrate converts into nitrite. So, NO3 minus converts into NO2 minus, and this NO2 minus converts into ammonia by nitrite reductase. So, also I have mentioned in the previous class that the first step, nitrate to nitrite, is a cytosolic reaction. So, that means this is carries over in the cytosol of the plant cell, whereas the next reaction is a plastidic reaction, and this takes place mostly in the photosynthetic plastids. The first step requires one NADPH and the second step requires 6 molecules of reduced ferredoxin. So, today mostly we discuss that how and why the nitrate reductase play a very key role in regulating the rate of nitrate assimilation or nitrate to ammonia conversion. This enzyme, look at this enzyme structure in the down, it is nitrate reductase, and this has 3 domains. The first domain is called FAD domain or FAD binding domain. Second one is a heme binding domain, and third one is a molybdenum cofactor domain. Please note this co is not cobalt, but cofactor. This is a protein with 3 domains, and the domains are linked with a linker, and these linkers are called hinge 1 and hinge 2. So, this enzyme can bind NADPH, and it can convert NADP, it can bind NO3 minus and generates NO2 minus. So, it transfers electron from NADPH to NO3 minus. So, this is the structure, overall structure of the enzyme, and this enzyme works as a dimer. So, you can see the structure here. This is MO-CoO, molybdenum cofactor, which interacts with the other half and makes a functional dimer structure. And the right hand is the how proteins are assembled in the ribbon structure of the protein. You can see here the FAD binding domain is shown in red, the heme binding domain shown in light blue, and molybdenum cofactor binding domain is green, and their interacting phase in yellow. So, you can see this yellow is between 2 greens, which are molybdenum cofactor binding domain. The next enzyme is nitrite reductase. It is a plastidic enzyme. This plastidic enzyme has again 2 domains. One is a ferredoxin binding domain, one is a serohome, and this enzyme uses reduction potential of ferredoxin, and that electrons from ferredoxin is transferred to nitrite to convert ammonia. So, if we look into now the energy requirement in these 2 processes, one is happening in the cytosol, the first reaction, second reaction is taking place in plastids. Now, these reactions overall depends on ferredoxin. Ferredoxin can be generated very well in photosynthetic plastids in the photosystem 1, and In addition, pyridoxines are also generated in non-photosynthetic plastids by the process of oxidative pentose phosphate pathway. So, there are ways for generating both of them. So, it is possible to use both glucose or carbohydrate as a source of energy, and in addition the time of photosynthesis where the light energy is being used in the photosynthesis to generate ATP as well as pyridoxine. So, now let us say that what is the contribution of already prepared carbohydrate versus the photosynthetic energy. So, here you have to see the economy of a living cells. When a living cells uses photosynthetic energy to make carbohydrate and to again digest carbohydrates to use ATP and pyridoxine to generate energy. In another way, one can have the photosynthetic electron, photosynthetically generated electrons which can be used for reduction. So, theoretically both are possible, but practically one can easily imagine that when photosynthesis is going on during the light time, plants can acquire large amount of energy through photosystem which can be used directly. So, we will look into these two situations, and you can see in this photosystem electron transport chain. This is the chain where photosystem 1 you can generate reduced pyridoxine. And this is the source of pyridoxine in non-photosynthetic tissue, which is also called as oxidative pentose phosphate pathway. So, some of you, if you want to read more, you can read any of the biochemistry books for this part. Now, coming to the main topic of discussion today, which I want to initiate, is how and why this regulation of nitrate is very important. The most important aspect, what actually also I have discussed in the previous class, is nitrite is very toxic. So, plants cannot accumulate nitrite above to a very small amount of quantity. So, what should be the mechanism to deal with such toxicity? So, there are some mechanisms which I have listed here. One is nitrite reductase is the second enzyme is abundant, whereas the first enzyme NR is rate limiting. So, NR amount is low, so second is produced at the lower level. Second is NR is regulated at the transcriptional level, and this nitrate assimilation is favored during light period to ensure NIR reaction. So, I have listed out 3 or 4 of them, which I will discuss one after another. Now, let us look into this basic concept how regulation should take place. See, I have drawn here 3 letters A, B and C. Now, say enzyme 1 is converting A to B, whereas enzyme 2 is converting B to C. So, in such a scenario, suppose B is very toxic, and this case like nitrite. So, you can say nitrate, B is nitrite, C is ammonia. So, if B is very toxic, naturally the system will be made in such a way that you have much higher amount of enzyme B compared to the amount of enzyme 1. So, the concentration of enzyme 2 or activity of the enzyme 2 must be way above than enzyme 1, and that is what is in the case of plant 2. So, nitrite reductase is very higher amount. So, again you can see here that nitrite reductase should be in a very higher amount in compared to the nitrite reductase. This is a paper, I have given out a heading of a paper. This paper was taken from a journal named Plant Physiology. The paper was published in 1976. So, it is about 40 years old research, but it is one of the very good paper that discussed how light is important in regulating nitrate reductase activity. You see, I have discussed a few minutes back that a plant can use both photosynthetic energy as well as glucose metabolism energy. So, this paper have discussed whether light plays the more predominant role in nitrogen assimilation compared to the glucose metabolism. Look at this graph here. This graph is taken from this paper only. Here the experiment was done like this. They have taken barley seedlings and these seedlings were kept in 7 days for under dark. So, when you keep 7 days in the dark continuously, photosynthesis was almost stopped, and during that period most of the glucose amount will be almost used up for the normal metabolism purposes. Then the plants were transferred into light, and after putting in the light, the scientists measured how much was the nitrate reductase activity in terms of production of nitrite per gram of fresh weight. So, in the y axis you have nitrite micromole per gram fresh weight, and the x axis you have incubation time. The time starts from keeping the light. Remember, these plants have very little amount of glucose because they are already kept for 7 days in the dark. Now, you can see soon they are transferred in the light, the nitrate reductase activity, the nitrate reductase activity started going high from 0 onwards and started going up. So, you can see here that nitrate reductase activity started growing up as it was kept in the light. Now, you can, if you look into this graph only, you see how the graph is taking a plateau. Now, that indicated that either nitrate was reducing or energy was reducing. So, to know that, what they have done, where this arrow is mentioned, they have added extra nitrate. So, this potassium nitrate is now used as a substrate, and these plants can now steadily make nitrite. So, this line shows the rate of nitrite production over the period of time. So, this is a clear cut suggestion which says that light energy can be used directly for nitrate reductase activity or for the production of nitrite. The same paper also had a very interesting data. That data I have drawn into a graph to show how much is the contribution of glucose versus how much is the light contribution. So, what they have done, again the experiments are very similar. They had kept the seedlings for 7 days in the dark, and then they have measured the nitrate after 24 hours of treatment. Look at the treatments. Here it is dark and no glucose, and then dark but glucose, and in the last one is the light but no glucose. Now, look at the rate of nitrate reductase activity. If you compare between the first block and the second block, the difference is only glucose. you see here there is this bar is bigger than this bar. So, there is an enhancement of nitrate reductase activity due to presence of glucose. So, this is what I spoke about the oxidative pentose phosphate pathway mediated ferredoxin contributing to nitrate reductase activity. So, there is a substantial amount of nitrate reductase activity increased by only glucose. However, if you give light, now look at this versus this, no glucose added, but light was added, and in the light the amount of nitrate activity goes very high. That suggests that if you have glucose or light, plants would preferably use light energy to make nitrate or to make nitrite from nitrate. So, I hope these experiments are quite clear to you, and to summarize these two experiments, I should say that plants use photosynthetic energy or plants prefer photosynthetic energy in compared to the metabolic energy for generating reduced ferredoxin for nitrate reductase activity. So, this is now I will discuss about two very interesting aspects. Under a normal day, suppose just think about any day, the sun rises in the east, slowly gradually sun goes up, and you can see the light of the sun starts falling on the ground even much before you can actually see the sun. So, that is what you called as dawn. So, there is a dawn, pre-dawn, and slowly after the dawn in the morning, slowly the sun rises and becomes very bright. Now, a few minutes back what I told you that nitrate reductase enzyme is under a light induced conditions. I will explain to you that also very soon. So, what I told you that plants prefer photosynthetic energy to make nitrate, to use nitrate. So, naturally the enzyme nitrate reductase is a light inducible enzyme. That means the gene will be expressed only when there is light. So, during bright sunny day there will be production of nitrate reductase enzyme, expression of the nitrate reductase enzyme, and at the same time there will be large amount of energy generated. So, that is what happens during a bright sunny day, and gene is expressing, protein is expressing, there is enough energy by the light. So, there is no problem. Now, if you think about a natural evening again takes place slowly over a period of considerable long time, say half an hour to 1 hour. During that slow period transition, say 2 hours from during the dusk time, slowly the nitrate reductase production will be stopped, the mRNA will degrade, the protein will degrade, and then the entire reaction will be shut off. So, that is not a problem. During normal day, production of nitrate reductase to its degradation is very controlled, that is not a problem. However, situation comes when there is a sudden shadow on the field. Like for example, you have a bright field, which is I have shown in the right side here, you can see there is a bright sunny day, and suddenly the field is covered with the cloud, and the cloud is very big, which is likely to keep it covered for long period. And another situation comes almost daily in the hilly region. In the hilly region, due to the shadow of the hill, a part of the plateau gets shadow even in the middle of the day. Like if you look into this picture, this is a very bright middle of the day, but this part suddenly gets shadow. So, whereas nitrate reductase enzyme is being synthesized over here, it will make nitrate reductase, but here it will make nitrate reductase with the protein, but it cannot convert into nitrite, convert into ammonia, because enough reduced pyridoxine or enough photosynthetic energy is not there. So, I will explain these two situation with the cartoon here. Look at this leaf. This leaf is receiving sunlight. So, is this leaf is photosynthesizing, this leaf is making nitrogen. Now, say this is one cell of the leaf. In one cell of the leaf, this is the nucleus. In the nucleus, you have DNA. This DNA, for today's discussion, assume this is nitrate reductase enzyme. So, now when light falls on the nitrate on the plant, this nitrate reductase RNA will be made. This RNA eventually will be transported out and then translated into protein. So, this is the process of transcription and then exportation to cytosol, generation, translation of protein. This overall step, starting from getting the sunlight, to production of protein is done in minutes time. So, it may take a few minutes time to generate large number of protein as soon as the lights are turned on. At the same time, if you look at the photosynthetic rate, the moment the light comes, it may take hardly a millisecond to turn the photosynthesis on. So, this is the reaction turning on in minutes, turning on a photosynthesis in seconds. So, in a natural day, there is no problem at all. Now, what happens during the evening? In the evening, this mRNA would be degraded, broken down, the protein will be broken down or degraded, but this process takes hours. So, this degradation of protein or degradation of mRNA, usually they are defined by their half life and it will take hours to do so. Again, in a natural day, that is not a problem, because even if you do not have the nitrate reductase, that is not a problem. Only problem when it is accumulates to a high level. So, suppose in a day, this is middle of the day. Suppose this is the middle of the day, plants are synthesizing very happily and suddenly it becomes shadow. So, when there is a sudden shadow, you will see for hours there will be more and more RNA and there will be more and more protein. Which protein? This is NR or nitrate reductase. So, when there is more nitrate reductase, but there is no light, then plants cannot utilize the Ni or nitrite. So, if plant cannot use the nitrite, it will be toxic. So, plants should have a mechanism to stop it. So, this is what actually I have also tried to draw here. Nitrate to nitrate, nitrate to ammonia. In sudden dark, NIR is non-functional, but mRNA and protein will continue to work for a few hours. So, naturally in a sudden dark, there is a possibility when this will accumulate, unless the nature takes care, and naturally nature takes care of everything. So, you will find what a beautiful mechanism the plants have to reduce that effect of sudden shadow or sudden darkness. See here I have drawn this again, this protein which you have seen it before. This is the enzyme complex, this has one hinge 1, hinge 2, and this is the protein complex, and this is what happens when there is a sudden shadow. And I will essentially, this is a called post-translational regulation of enzyme. Here I should say post-translation regulation of NR activity or nitrate reductase activity. Post-translation means the protein is made, it is translated, then there has to be a modification. So, I will discuss with you about the modifications which are important here. There are large number of different type of modification takes place in the proteins. They are commonly called as post-translation modification. One of the very common modification is phosphorylation. Say this is a protein, any protein. This protein can get attached to a phosphate group, and here phosphate donor is ATP. This is an ATP. This ATP has a 3 phosphate, that is why it is called triphosphate. Out of these 3 phosphate molecules, one of the phosphate can get attached to the protein. So, protein become phosphorylated protein, and by losing one phosphate, ATP become ADP or adenosine diphosphate. So, there are 2 phosphates now. So, this is one of the common kind of modification that takes place in many proteins by protein kinase. Say what happens in this case. This is the structure of the protein nitrate reductase with the 3 domains, and this is an active protein. In plant there is an enzyme called nitrate reductase kinase. So, this enzyme works on nitrate reductase enzyme and can phosphorylate. See, look here, this is the direction drawn here. This uses ATP and this converts into ADP, and this is the process which is promoted by a signal. This signal can be a dark signal or other stress signal, usually activated by calcium and effectively suppressed by light. So, when light is not there, then only the reaction goes on and nitrate reductase gets phosphorylated. All the kinases, if not all, most of the kinases in the plants or animal or any system are coupled with a phosphatase. So, kinase adds the phosphate and the phosphatase removes the phosphate. So, this is a reversible reaction. So, it can happen, this addition of phosphate can happen to many amino acids like serine, threonine, tyrosine, histidine, aspartate and so on. However, in this particular case of nitrate reductase, something very interesting happens. This site is marked here is a specific serine molecule. Serine is an amino acid, and this serine is in a context where you can see there are 2 lysines, K is lysine, another K lysine, serine, X and then proline. So, this is a series of amino acids which are present in this area, and here serine get phosphorylated. So, this phosphorylation of serine molecule becomes a different type of phosphorylation where it attracts other protein. The name of that protein is called 14-3-3. So, I will just introduce to you what is 14-3-3. So, some of you may know about 14-3-3, if not you can type in Google 14-3-3 protein, you will find vast amount of information about this protein, and you will know that this has a role in many diverse function of living cells, both in animal and plant. This 14-3-3 are small protein that can bind as a dimer in a protein where the specific serine molecule is phosphorylated. So, when a certain molecules like what I told you in NR get phosphorylated, that becomes a binding site for 14-3-3. This 14-3-3 binds in duplicate and that can inactivate a protein. This 14-3-3 was first time discovered by someone, here is written Murray and Perez in 1967. This Murray and Perez, they were studying the proteins present in bovine brain. So, they have taken the brain tissues and isolated protein from the brains, and then they fractionated. The proteins were first column fractionated and then they are separated on the basis of electrophoretic mobility. Okay. So, this 14-3-3 would indicate that this has the 14th fraction having RF value of 3.3. So, many proteins have been found later on to be very important in this from this assay, but 14-3-3 name has been not changed because large volume of work was carried out before identifying or before knowing what 14-3-3 does. So, the name remained as 14-3-3. So, now see what happens to this protein. This is the NR or nitrate reductase. This nitrate reductase undergo phosphorylation by phosphate kinase. Now, look the interesting point. When it is phosphorylated, it is not inactivated, it is still active. I have shown the same thing over here also. You can see here, this is an active protein and even after phosphorylation the activity is not lost, that still remain active. However, when this active protein binds with 14-3-3, so there is a 14-3-3 dimer, this is one protein, another protein dimer. This 14-3-3 when it binds here, this becomes inactive. So, now what happened, you see what I told you, this degradation of protein might take hours, mRNA might take hours. However, phosphorylation can take in minutes. So, whenever there is a sudden dark or sudden cloud in the sky or in a hilly area, there is a shadow in the middle of the day, this NR will be phosphorylated within a minute, within some minutes, and then it will be inactivated by 14-3-3. 14-3-3 is quite abundant. So, naturally this protein will not work. So, there is no toxicity is of the NIR. I just told you about this story essentially in details to tell you that, or to help you to appreciate the development what takes place in nature, how carefully nature blends every aspects of life so that life functions very smoothly. And this is what exactly in a case where I have shown you how nitrate reductase itself is induced by light and degraded over slowly in a natural day, but inactivated very promptly when there is a sudden darkness. So, that is what the beauty of the nature is. So, this is the again a summary of the my nitrate reductase slides, what I have told you about this. Okay. So, now I will talk to you about what happens when nitrate is there in the cell. So, nitrate is taken by the cell and this nitrate is taken up, and I told you already that this nitrate converts into nitrite, and this nitrite go to plastid where you have huge energy available, and this nitrite converts into ammonia, and this ammonia converts into glutamate. So, this one is the GS go get cycle I have told you in the previous class, and this is how the nitrate assimilation takes place. This is the ammonia go get cycle which I have already discussed. So, otherwise you can also look at my lecture which I have delivered in the last class. So, in brief, the glutamate takes ammonia and converts into glutamine, and this glutamine again reacts with another molecule of alpha ketoglutarate, another molecules of glutamate, and can convert into 2 molecules of glutamate. So, now the last part of my this nitrogen assimilation as such, I will tell you about how plants absorb nitrogen, because without absorbing it cannot really convert into anything. So, many of the students of today, you may be knowing what are called pumps, carriers and channels. If not, please have a printed book, that is really good to read, or if time permits also I will take a class specifically, because there are many things to discuss. In brief, let me tell you, the molecules must transport from one part of the cell to the other part of the cell, or from the outside of the cell to the inside of the cell. This can happen in the favor of gradient, like where amount is more, it can come passively by diffusion, this is called passive, or by the assistance. The assistance is made with the help of energy. There is diffusion, what I told you, from the higher gradient to lower gradient is facilitated by something called channels. These channels have a gate, the gate is opened by the signal, and when the gate is opened, it diffuses very rapidly, large number of molecules come together. But most often in a cells, the pumping must be taking place against the gradient, or lifting, like water lifting to the roof, and that requires energy. So, there are 2 types of system that can do it, one is called carrier, one is called pump. So, I will tell details about it, but to briefly, there are molecules which can bind with say ATP, and that energy can be used for transport, which are mostly used as a pump called pump, and there are carriers where it can use proton motive force mostly, or many times without proton motive force exchange of molecules, like one molecule will come from one side to the other at the exchange of another molecule. So, it is like exchange of molecules. So, based on this exchange molecules, these carriers are broadly called as symporter and antiporter, and say most often the currency is proton here, like because you see the protons are abundant is true, but at the same time every cell has the mechanism to generate protons in a specific compartment of the cell. Like for example, here I have drawn a membrane here, and this is outside the cell and this is cytoplasm. Say molecule A can come inside with the help of a proton. So, proton and molecule A both will come together, but if it comes, what will happen? This part will be acidic, because see more proton means more acidity. So, naturally there has to be another pump to move it out. So, this is in contrast, actually this is called sim protar. Sim is similar. So, the direction of the proton and direction of the molecule being carried is the same. This is called sim protar. In contrast, there is an antipotar. In case of antipotar, the direction is opposite. So, here proton is coming from outside to inside, whereas the molecule is going from inside to outside. So, this is a two-way road and this is kind of a one-way road, where both the vehicles come in the same direction. Here one vehicle goes one way, other vehicle goes other way. So, this is called antipotar, and these importers and antipoters play very important role in nutrient uptake by plants and other system. So, if you look at here like ammonia and nitrate, I told you both of them are used by the plant as source of nitrogen. But if you look here, the ammonia is positively charged, but nitrate is negatively charged, and both of them are carried inside with the help of proton pump, proton motive force. Look here what happens in case of ammonia. Ammonia is a positively charged molecule. This ammonia can go in at the cost of one proton. So, the proton goes out and ammonia goes in. So, this is an antipotar. And what happens actually in case of plants especially, they try to keep this cytosol pH is almost around 7, which is a normal pH. Outside pH is low, it is around 5.8. So, here hydrogen concentration is more, here it is less. So, ammonia taking in actually requires hydrogen to be pushing out against the gradient by spending the energy, and that can be done by a pump kind of thing which is called as a proton pump. So, it is shown over in the right hand. In the right hand you can see, look here, this is nitrate. This nitrate comes in and at the same time it carries in two molecules of proton. So, because protons are quite high, it is easy to carry with the help of a proton level. So, one molecule of nitrate which is single negatively charged brings in two positive charged proton. So, once this is carried in, then what happens? There is an additional positive charge. So, plant cell must retain the neutral charge in all the time. So, what it does? It takes one molecule of proton and this proton is thrown out by the proton pump. So, overall charge of the cell remain same. So, this is what the main difference by how ammonia and nitrate is taken up. Now, look at the quite interesting thing between these two. In case of ammonia, more and more as ammonia is taken, more and more proton is thrown out. So, if only ammonia fertilizer is used, soil is going to be acidic, because soil is getting reached with more and more of proton. In contrast, every molecule of nitrate when it is taking, it is actually losing one molecule of hydrogen ion. See here it is taking two hydrogen ion and throwing out one hydrogen ion. So, for every nitrate, soil is losing one proton, or other words, soil is actually getting alkalinized. So, in case of ammonia, soil become acidic. In case of nitrate, soil become alkaline, and that is the one of the reason or maybe most important reason why most of the fertilizers comes in a combination of ammonia and nitrate. So, when it comes in a combination of ammonia and nitrate, then the soil pH remains balanced. So, it is neither go too acidic or too alkaline. There are many transporters available, and many of the transporters have been identified from plant system, but it is quite difficult by the way classical genetics is find the genes, because every say nitrate or ammonia, plants have large number of transporters. So, even if you mutate 1 or 2, you are not going to get a phenotype. To bypass this problem, scientists have used a system where a screening is done based on chlorate. You see, this is a chlorate structure where one chlorine and ClO3 minus. The ClO3 minus atomic structure is similar to nitrate. So, the transporters that can take nitrate in also can take chlorate in, but this chlorate is very toxic, and chlorate cannot be converted into any other non-toxic form inside the cell. So, a plant which is mutated for nitrate uptake will only be able to survive, but not the natural one. And this is how many of the transporters have been identified. So, I am not going to go details about the transports isolation and all. This maybe you can study at the higher MSc level or so, but I am just wanted to give you a flavor that how transporters are isolated, and especially the problem comes when there are many genes work for the same phenotype. So, there must be an assay system for identifying the mutants. So, this is how by now I am almost done what I wanted to tell you over the last 3 classes, that how nitrogen is used up by the plant in a process of assimilation. So, if I now just go and summarize this over the last next 2 minutes available to me, I would say that nitrogen is very important. Nitrogen is important for phytohormones, for pigments like chlorophyll, it is important for the vitamins, it is important for DNA, it is important for protein. And this nitrogen is acquired by plants from soil mostly. Nitrogen is there in the air, is not useful. The elemental nitrogen converts into nitrate or ammonia by bacteria, and bacteria can do it alone or in combination with a plant can convert into ammonia, and this nitrate and ammonia are taken up by plants, and once they are taken up, if it is nitrate form, plants converts to ammonia form, and this ammonia gets incorporated into glutamate, and this glutamate is amino acid. It's also a part of a metabolite which is abundantly available called alpha ketoglutarate. This alpha ketoglutarate combines and comes into the picture where it can readily recycle ammonia to many different molecules, and that is how the assimilation of nitrogen takes place. And plants have very large number of transporters to transport nitrogen from soil to inside the plant, and in addition to what I have discussed also today is that plants have the wonderful mechanism to regulate its assimilation, regulates its conversion, so that plants never accumulate to a high level, so that it becomes toxic to itself, and that is the process by which it survives itself and helps entire living kingdom to survive. So, thank you very much. I hope you have liked it.
With this note, thank you, sir. Thank you so very much for giving your precious input this particular lecture. Dear friends, if you want to access this lecture, then you can access it with the help of YouTube, as very soon we are going to upload this lecture on YouTube, with the help of which you can study the content which Dr. Nandi explained many number of times, any number of times you would like to see. And dear friends, if you have any queries or feedback regarding this particular lecture, then you can mail us at info.cec@nic.in. We would be meeting again with a new topic. Till then, take care. Bye bye. Thank you, sir. Thank you so very much.